Dr. Baliga here. Welcome to today's episode of Got Knowledge Doc podcast, where we explore groundbreaking discoveries in genetics and the implications for science and medicine. Today, we are delving into the fascinating world of genomic imprinting. This remarkable phenomenon, discovered in 1984, fundamentally changed our understanding of gene regulation and inheritance. This podcast was derived from a News and Views article in Nature Journal published on 14th May 2024 titled "The Phenomenon of Genomic Imprinting was Discovered 40 Years Ago", authored by Dr. Anne Ferguson Smith and Dr. Marisa S. Bartolomei. The discovery of genomic imprinting. In 1984, a series of pioneering experiments by Surani, Barton, and Norris, as well as McGrath and Salter, revealed that the expression of certain genes in mammals depends on whether they were inherited from the mother or the father. This discovery was a breakthrough in understanding how gene regulation works. Unlike most genes, where both copies are either turned on or turned off, imprinted genes have one copy active and the other silenced depending on its parental origin. Landmark experiments. The key experiments involved manipulating mouse embryos to have either two maternal or two paternal genomes. The results were clear. Embryos with two maternal or two paternal genomes failed to develop normally. This indicated that both maternal and paternal genomes are essential for normal development. These findings highlighted that maternal and paternal chromosomes are not functionally equivalent but carry unique imprints or epigenetic modifications that determine gene expression. What are the mechanisms of imprinting? So, what exactly are these imprints? They are epigenetic modifications, primarily DNA methylation, established during the formation of eggs and sperm. After fertilization, these imprints remain, ensuring that specific genes are either expressed or silenced throughout the organism's life. Each imprinting control region, or ICR, in the DNA has unique methylation patterns that mark the parental origin of the gene. Identification of imprinted genes. The journey didn't stop with the discovery of imprinting. In 1991, the first imprinted genes, including IGF-2R, IGF-2, and H19, were identified. These genes play crucial roles in growth and development and are only expressed from one parental copy. The discovery of these genes led to the identification of many more imprinted genes, with around 100 to 200 now recognized. These imprinted genes are often clustered together in the genome and include both protein coding and non-coding RNA genes. What are the implications for human health? Genomic imprinting has significant implications for human health, particularly in understanding and diagnosing imprinting disorders such as Prader-Willi syndrome, Angelman syndrome, and Beckwith-Wiedemann syndrome. These disorders arise from mutations or epigenetic alterations in imprinted genes, often resulting in complex developmental and metabolic issues. Researchers are exploring these therapeutic strategies to reactivate the silent copies of imprinted genes to treat these conditions. What is the broader impact on epigenetics? Beyond imprinting disorders, The study of imprinted genes has provided valuable insights into epigenetic regulations across the genome. It has helped scientists understand how stable epigenetic states are maintained and how they can influence gene expression over multiple generations. The principles learned from imprinting studies are now applied to broader epigenetic research, offering new perspectives on gene regulation, development, and disease. In conclusion, the discovery of genomic imprinting 40 years ago opened a new chapter in genetics, highlighting the intricate dance between our maternal and paternal genomes. As we continue to unravel the complexities of epigenetic regulation, the legacy of these groundbreaking experiments in 1984 reminds us of the profound impact of scientific exploration. This podcast was derived from the Nature article mentioned earlier. Thank you for joining us on this journey into the world of genomic imprinting. Stay tuned for more episodes of Dr. Baliga's God Knowledge Doc podcast, where we continue to explore cutting-edge medical and genetic research. If you enjoyed today's episode, don't forget to subscribe and share it with your friends and colleagues. Follow us on Twitter and LinkedIn for updates and join the conversation. Until next time, this is Dr. Baliga's God Knowledge Doc podcast.